Chapter 2, Basic Principles and Concepts, Parasite Systematics, Ecology and Evolution. Parasite Ecology, Part 3. When it comes to parasite populations, we want to have what are referred to as quantitative descriptors. Okay, qualitative descriptors will be, high levels of, low levels of, seem to, impact, greatly impacted, had no impact, little, minimal impact. Words that are very squishy, okay? We don't like squishy words, okay? We want numbers. We want to be able to sit there and say, this is exactly what we counted, and this was the number. We counted it this day, 6 months ago we counted it that day, and here is the numbers for both days or for both animals, and now we can compare, okay? So when we go and we look at qualitative descriptors, as they're called, these are important from the medical standpoint. People, the individuals in the medical fields, the doctors, the nurses, the scientists doing research, the health department, the veterinarians, the wildlife people, whoever. They want to have numbers of who's getting sick, why are they getting sick, and how are they being exposed, okay? So they're going to focus on prevalence, how many people, where, incidence, what are the total number of animals, total number of people that are getting sick, the abundance being, out of the population. What's the populations, what populations are we looking at? What's the percentage of? And things like that. When you look at table 2.1, as you see here, some of the different nomenclature, which is things that we're not going to, we don't truly use, except when we're discussing epidemiology, when we're discussing the who and the how and the why people are getting sick, the sampling unit, the density, the intensity, the abundance, the aggregate. These are going to be terms that allow us to describe specifically who's getting sick, how are they getting sick, why are they getting sick, and what's the means we use to count the number of people sick versus the number of people who were exposed not getting sick, and out of the total population of individuals that we counted. Okay, when you look at populations, you need to take into account this idea of macro versus micro. Macroparasites, microparasites. Macroparasites, as you see here from this, definition that I've taken from the textbook, large, that parasites that do not multiply on or in a host. What does that mean? Well, these are going to be large organisms, multicellular, that what you ingest, that number of parasites is going to be the number you have in you. If you ingest the larval stage of 4 cestodes, remember the cestodes are the tapeworms, you're going to end up with maximum 4 tapeworms in your large intestine. In your large intestine, they are not going to go through asexual reproduction and one becomes 2. You ingest 4, maximum number you're going to get is 4. Okay? What we find is that the macroparasites, because of the way they spread in the populations, and they call this aggregate, that there's going to be minimal to large number of parasites and a minimal to low number of hosts. So tapeworms are not going to be spread throughout a population where, 50%, 75% of a population has tapeworms, it's actually going to be much, much smaller. 5, 10, 15% of a population of humans within a given area will have tapeworms. And those that do are going to have a balanced number. They're not going to be overwhelmed. It's just one of these weird aspects about the evolution of the macroparasites. They do not want to, apparently evolution is set up so they don't overwhelm and thus have the chances of not only killing their host, but killing a large percentage of a population. Because if a large percentage of a population dies off, well, the health of that population begins to decrease, and that population, given enough time, it will eventually end. Mirror image of this, on the opposite side of the coin, or however you want to think about it, is the microparasites. Microparasites are going to be the ones that, once you've ingested them, you ingest one, come back a couple months later, and you've got a few thousand, a few hundred. Some of those stay, some of those pass out with the feces. So the micro are going to spread through a population very easily. You're not only, the numbers you ingest or the numbers you get exposed to are not going to be related to the number that you're going to end up with, because that population is going to continue to expand and increase within the host, can overwhelm the host. So when we go and we look at populations, we go as the epidemiologist, as the ecologist, parasitologist, wanting to study not only the life cycle of the parasite, but determine the host, definitive, intermediates, and whatnot. How's the spread? What's the vectors? We want to look at the population structure. Who's getting exposed? Who's getting sick? For how long? What separates the first two? Exposed, sick. When it comes to, those getting sick, severe, minimal impact, things like that. So we begin fractioning out, based on a whole list of criteria. Age, sex, socioeconomics. And I know I've mentioned socioeconomics here in the previous lectures, and I'll mention it again. Turns out socioeconomics does come into play based on, first world, third world. Within the first world, who's rural, urban. In the urban, are we talking a higher end, San Francisco, lower end, Detroit? Okay, things like that also come into play on who's getting exposed and to what. So we can go through, and as you see on this figure here from the textbook, we can go and we can begin separating out dates, when was samples taken. We can, how many people, how many hosts. Of the people that are getting sick, what's the parasite load? Few, heavy. We can begin breaking it down. You look at this figure here, on the y-axis, frequency. How many percent of people, of those sick? The different shadings, denotes years. And you can begin to see that, low to minimal, 0 to 10 cysts or parasites per host. And there are some that gets up to 50 or more. And as those numbers increase, what's the parasitic load? We see that all 3 years, the higher the parasitic load, the fewer people that are, the fewer the people there are, the fewer the hosts. The majority of the hosts are going to be at, 0 to 10. 0 to 10 parasites. 0 to 10 parasites has about high 60s, low 70s% of the population that was sampled. When it comes right down to it, we see that there is an interplay, a relationship between the parasite and the host. Okay? The host has something the parasite requires. But what are those requirements? Well, let's think about what the parasites are. The parasites are eukaryotic, most of the time multicellular organisms. Some are going to be single cell, depending upon where they are in that life cycle, which developmental stage, or just what the species is. You take the parasitic protozoa, they will always be single cell. All of them, no matter if they're single cell, multicellular, are heterotrophic. Okay? Heterotrophic means that they have to get their nutrients, their carbon sources, from outside the body of the parasite. They have to bring it in, internalize it in some way. We internalize by eating. Some of the parasites eat. Others are small enough, thin enough that they just absorb it through their integument or absorb it through their cell membrane. Because of their ecological niches and their nutrient requirements, their carbon source requirements, the parasites have all evolved to, maximize their ability to get the nutrients in. These abilities, these adaptations are going to be different for each of the parasites, each of the different classes, each of the different genii. You take the cestodes, the tapeworms, okay? Tapeworms have no internal structures for digestion. They really actually don't even have digestive tissues. Why? Because they are so thin, they are flattened, wide but flattened worms. They're flattened in such a way that the surface area is maximized to volume of the worm, so that they can absorb nutrients here, and those nutrients, that concentration of nutrients, will be able to diffuse throughout the entire proglotid, so that all cells that make up the section of that tapeworm will be bathed in nutrients. This is on, the flip side of, say, the liver flukes. Liver flukes are going to have a rudimentary gastrointestinal tract. They will take in cells from the liver. They will take in bile. They will take in nutrients that way. It'll go, some of them have a true digestive tract. Some of them have just a pouch, depending upon which flukes you are talking about. But whatever those stresses are, whatever those adaptations are, we see that the parasites, over time, big T time, okay? Big T time, the parasites have exploited the hosts, okay? Those parasites that just kind of, sort of got along, selected against. Those parasites that maximized, maybe even pushed the boundaries of just how much they could take, of just how much of the niche within the host they could fill. Those are the ones that did just a little bit better. Those parasites were able to do just a little bit better. So their lineage, their genetic abilities that allowed them to push the boundaries, stretch it just a bit, not go too far, because they go too far, death of the host, death of that parasite lineage. Those that maximized it, just pushed it a bit, are the ones that over time won out. They kept going and going and going. Other ones died off. When you look at this figure from the textbook here and you see a rudimentary life cycle, the definitive host is the bird. The sexual reproduction occurs in the bird in the gastrointestinal tract of the bird, specifically. The bird's going to pass out eggs. The eggs are going to hatch. The oocytes are going to hatch the fresh water. And the different larvae, depending upon the species, are going to swim and invade a snail. And they're going to become a microparasite of the snail and begin going through asexual reproduction, mitotic division, increasing in number within the snail, and then as they divide and the population increase, they will start going through larval stages. They will either exit out or be eaten by the fish, and you can see, depending upon which of those circles you go through, direct to a snail, to a mollusk, to a small crustacean, or small, some small whatever. I can't think of any of the small little things that swim in the water that eat, that eat the bacteria. Well, it turns out the little larval oocyte that hatches is going to be just a little bit bigger than the bacteria, but still small enough that some rotifer will come and eat it. The rotifer gets eaten by something bigger and bigger and bigger until it gets eaten by crustacean. Crustacean gets eaten by the fish. So whether it's the snail, the mollusk, the crustacean, it's going to be getting eaten by a fish. More microparasitic, invasion here gets into the musculature of the fish. There, it's going to sit, its wait until it gets eaten by a bird. Oh my good Lord. Think about how many millions of years this took of slow adaptations, of slow little incremental steps, so that certain lineages of these primordial species of whatever parasite could somehow close that loop, close that circle. And think of the how many hundreds or thousands of unknown species, unknown lineages of this primordial species that had to have died off because they either pushed the envelope too far with, parasitic loads or, things like that, and killed off hosts too quickly, so that lineage died off, or they didn't push enough, they didn't adapt enough. They hit a dead end. Well, fast forward millions of years and they're no longer here.